My Aunt Rose came home from work that day as if she had seen a ghost. That's the way my cousin described it. And she said, "Mommy, what's wrong?" And she says, "I've just seen a miracle."
5 ways that glycine could potentially help your brain.
I'm a woman in my 30s, and I've had 3 kids, and I've had horrible migraines like almost daily for years and years and years, and my life's been hell, and they've gone away. So migraines are definitely due to inflammation. People say, "Oh, well, I have a complete diet." If you don't have enough glycine in it, no, you don't. That's really a key discovery. G protein receptor 158, which was an orphan receptor, now they just found out it's a receptor for glycine. And they put him on a ventilator, and he was still alive, but barely alive, but he's completely non-functional. He's just like a dead man in bed. About a week later, I said, "How's your dad doing?" He said, "It's like a miracle." He said, "You're like an angel who came in the night."
Hi everyone, if you're feeling like you have brain fog all of a sudden, troubles learning or with your memory, maybe you're depressed or anxious or have other mental health symptoms, today we're going to talk with a favorite on the show, Dr. Joel Brind, author of the Glycine Miracle. He's an NYU and Yale-trained biochemist, a glycine expert, and we're going to discuss 4 or 5 ways that glycine could potentially help your brain with many different conditions and groundbreaking research that's not ours, but that Dr. Brind is going to help us interpret as a glycine expert regarding depression and other mental health issues, and which drugs are being developed that work with glycine and what that might mean for supplementation and using glycine in general. So welcome to the show, Dr. Brind.
Thanks again for having me on. My pleasure.
Love talking to you. And I cut you off on a story you were telling about your family member. I think it was your father's father's sister.
My father's sister.
Tell us about your experience. Yeah, tell us about this story. Yeah.
You may have heard or read the book or seen the movie One Flew Over the Cuckoo's Nest, which is about a state psychiatric hospital. In fact, it was Trenton State Hospital in New Jersey. And my father's sister she was not the chief psychiatrist because she wasn't a man. You had to be a man to be the chief, but she was like the real top of the food chain there in psychiatry. And my cousin, her daughter, Nina, relates the story of going back to 1953, when I was just a babe. In 1953 were the first clinical trials of the first antipsychotic drug called Thorazine. All the other antipsychotics are basically children and grandchildren of Thorazine. They start with that basic thing, and they've modified it and improved it and made it more specific. And lots of different varieties of antipsychotics out there. And they are miracle drugs. But even the first one, Thorazine, when they had the clinical trials of that in 1953, she said her mother came home from work, my Aunt Rose came home from work that day, as if she had seen a ghost. That's the way my cousin described it. And she said, "Mommy, what's wrong?" And she says, "I've just seen a miracle." What was the miracle? She saw patients of hers at Trenton State Hospital who were schizophrenics, who were basically useless, basically dead people living in straitjackets in rubber rooms. All you could do was sort of, try to take care of their needs and keep them clean and keep them, with nutrition, but they couldn't communicate. They were really not good in any kind of human interaction. They were gone people, the living dead. And when they tried Thorazine, all of a sudden, they were people again.
Wow.
And so these antipsychotics worked. And now, if you know anyone who has any of these major depressive disorders, schizophrenia, bipolar disorder, various other kinds of things these are very serious disorders. Now you say, "Oh yeah, my cousin, my friend, my this one, my that one, they have bipolar disorder. They're good people and they're fine." They're only fine because they take a cocktail of antipsychotic drugs and other drugs that their psychiatrists figure out on an individual basis what makes their brain function normally or close to normally so that they can actually function in the world as real people. So just, I got a lot of beef with the pharmaceutical industry, don't we all? They're all commercial entities chasing a buck, and that's going to run afoul of various ethical concerns and so on. But some of the drugs that have been developed are really, really miracle drugs. And that's just one story that I happen to know.
I think it's a great way to frame this conversation because like you and I said, we like the best of both worlds, right? I'm a very holistically inclined person. I wish nutrition was the baseline intervention for mental health disorders, for physical illness, for, you know what I mean? So that we could prevent all of these things. But I know what it's like to live with someone who is mentally ill. My aunt was bipolar. And I also know my mom, was a special education teacher, and will argue that drugs change lives and families and dynamics
Good one.
in profound ways when they need it. Yes, when used correctly. Not for everybody, but there is that. So I love that story, Dr. Brind. Thank you for sharing that. And just before we dive into the mechanisms by which glycine can affect the brain, just give people the high-level overview. Who are you? How'd you get here? Why do you like glycine so much? Yes.
Well, I started with a rather traditional training. I have my bachelor's degree from Yale in biology and a basic medical sciences PhD from NYU in specializing in biochemistry, physiology, and immunology. And for a long time, right through the 1980s, I was a professor of biology and endocrinology from 1986 through 2020 when I retired. Well, I'm doing more research these days now. I don't have a lab anymore. I'm not puttering around with test tubes, although sometimes I wish I could because, certain things are real easy if you have a lab. And when questions come up that can be easily answered with a lab, it can be frustrating when you don't have one anymore.
Yeah.
But be that as it may, I was actually in a medical research professional since 1972, so that makes it about 54 years. So I've been, working in this area, studying, teaching for a long time in 2007. When I first got on to glycine, and that had to do with the context of actually the metabolism of another amino acid called methionine, an essential amino acid. Glycine is often considered non-essential. We've since proven that not to be the case. But the critical finding in those research days, in the early 90s actually, going back to the early 90s, this was established group of people that I was working with at a private foundation in Hudson Valley in New York, was that if you feed rats a diet and they eat as much as they want, and it's just almost deficient in methionine, it has just barely enough methionine in it for them to survive, they actually thrived and lived about 30 or 40% longer. This group that I was working with, their primary focus on research was aging. How do we stop aging? How do we extend lifespan? All of that. So that was a very useful model. They first published that in 1993. And since then, it was reproduced in rats and mice and by many other labs, and it became really a thing. And anyone who doubts that can go into the aging research field and search methionine restriction. And you'll see that methionine restriction is a thing. So the question there was translational. Translational meaning, how do you take animal data, cellular data, test tube data, and make it relevant to the human condition? How do we extend our lifespan? How do we keep ourselves free of disease and so forth? The translational question that I had was then, if a methionine-restricted diet will add years to your life, how can we modify the human diet in order to simulate methionine restriction? And I say simulate or mimic methionine restriction because an actual methionine-restricted diet is something nobody would eat. Because all the tasty, wonderful foods, they're all full of methionine. All the meat and fish and poultry and all the delicious stuff. A vegan diet, it tends to be low in methionine except for certain vegetables. But for the most part, for most people, it would be very, very, very hard to follow. No dairy, no meat, and any of that. So the question was, what can we add to the diet which would help to clear out methionine? Because methionine is an interesting amino acid. It has been known for many years. In fact, anyone who studies biochemistry in graduate school or medical school learns about the methionine cycle. So methionine, as I say, many of your listeners may know about methylation. A lot of people who low methylators and all of that. Methylation depends on methionine. Methylation is a biochemical process whereby one carbon units called methyl groups are added to different compounds in order to change their behavior, their chemical function. So, for example, it's key in DNA modification, turning certain genes on and off, the methylation of DNA, methylation of compounds that may be hormone. For example, the conversion of norepinephrine to epinephrine is a methylation reaction. Making the acetylcholine is methylated in order to clear it, to get rid of it. Many drugs are cleared by methylation by the liver, so that gets rid of them. So that's what I was thinking of was, you know what, maybe we can get rid of methionine by treating it, since the liver actually treats it like a toxin when you have too much. So we all learned about the methionine cycle, the key ways, redundant ways that the body has to recycle and regenerate methionine so that it can get by in between meals. So it's an essential amino acid, but you need very little of it because your body can regenerate it and recycle it. However, when you have too much, like our typical diet, because methionine is rich in meat, fish, poultry, muscle meats, dairy, very high in methionine. You don't really do much of the methionine cycle in the hour or two after you eat. In the hour or two after you eat, your liver is actually doing the reverse. It's getting rid of methionine as fast as it can. So what I was thinking, where my mind was going in considering this question from a research point of view back in 2007, 2008, was what can we add to the diet which will make methionine get used up so that it can now stimulate a low methionine containing diet. First drug I actually thought of a drug called spironolactone, which is a relatively mild blood pressure medicine, which you take in doses of hundreds of milligrams at a time, 200 milligrams, 400, 600 milligrams a day. And the way that drug is disposed of by the liver is by methylation. So I thought, maybe we can put people on a regimen of enough spironolactone that would actually decrease the amount of methionine. Well, it really turns out to be not practical. We didn't actually try it. That was just a theoretical exercise. But of course, I was in the literature looking for ways that you can actually accelerate the clearance of methionine. So at that time, in the early 2000s, there was a lot of research that had been done on these amino acids and their metabolism. And it turns out that glycine, the main pathway that was elucidated, was a pathway centered around an enzyme called glycine and methyltransferase. All the enzymes that take that methyl group out of activated methionine is called SAMe, you may know as adenosylmethionine.
Yep.
The enzyme itself is called glycine and methyltransferase. That's interesting. Glycine, the normal, it's a very simple nutrient, actually should help the body to clear methionine, because that's what the body uses when it has too much methionine to get rid of it. And incidentally, it takes 2 molecules of glycine to get rid of every 1 molecule of methionine, one through a process called transmethylation and the other through a process called transsulfuration. It uses up ultimately 2 molecules of methionine. Well, so that should work, I thought. So the simplest way to test that would be in our laboratory animals. We were working with Fisher 344 rats. We're interested. The way to test that would be to just add certain percentages of glycine to their normal diet and see if it affected their lifespan. And lo and behold, it actually did. I presented the results for the first time in 2011 at the Federation of American Societies of Experimental Biology. That's a big, all topics in biology meeting that happens every year, and it's been happening for decades and decades. In fact, I found out after I published my abstract that it was on virtually the 60th anniversary of an abstract published at that very meeting in 1951 saying how glycine can ameliorate methionine toxicity in rats.
That is crazy.
So in that paper, we described how glycine actually does, and the strategy was, the hypothesis was, can we use dietary glycine, supplemental glycine, in order to help to clear methionine, and thereby the animals will live longer. And what happened is the animals did live longer. Not 30 or 40%, maybe 10 or 20%. But the interesting thing also is it did not affect the amount of methionine in their body, in their blood, or in their liver. They still had the same amount of methionine, but it made them live longer. So glycine helped the animals live longer, but something else was going on. It was not methionine restriction. And the hypothesis then switched to, well, maybe it's not methionine restriction, but that normal, quote, normal methionine maybe is too high, could be considered a toxic level, and that it's glycine, it's methionine toxicity, really, that which was getting rid of glycine, and that glycine was the important nutrient there that glycine was toxic to. So you see, that's very interesting. It took 60 years to come back to complete the yang of that yin at the Federation meetings. So anyway, what happened, what we realized then is that there's something about glycine. And at the time, well, even earlier, throughout the 1990s, there was some very, very seminal key research being done in a couple of different places. One of them was the University of North Carolina, a toxicologist named Ron Thurman, who has since passed away. He passed away actually in 2001, which was too bad because his group published the basic material which shows the main way that glycine works to stop inflammation, that it works through so-called what is so-called the classical glycine receptor. And it's the same receptor that is in the central nervous system, in the glycinergic pathways of the central nervous system, which is we're starting to get toward the topic that we had in mind for today.
Yes.
That's where that receptor, the glycine receptor, was first identified a long time ago.
So where specifically in the central nervous system?
Where? Usually in an inhibitory pathway. Once you get to the central nervous system, everything intersects. So it's very hard to say exactly what is doing what, but we do know that the classical glycine receptor is an inhibitory neurotransmitter. And
what does that mean for people who don't understand what an inhibitory neurotransmitter?
Oh, I got to get into neurotransmission. Okay, well, neurons are nerve cells, right? And the nerves are wires. They're like the wires between different parts of our body. So the wires aren't actually wires. They are long cellular processes, like a long extension of a cell. Like, for example, if you wiggle your toe may be 3 feet away, and the neuron, the ending of the neuron, where that stimulus to stimulate the muscle in your little toe emanates, is from a cell where most of the cell and the mitochondria and the nucleus and most of the cellular machinery is actually in your spinal cord. That's how long this. You say, wait a minute, cells are little. How could the cell be 3 feet long? Well, imagine the cell in a giraffe.
Yeah.
they may be closer to 30 feet long. They can. So cells are pretty amazing things. So yes, they're small, but boy, can those wires be very fine. And you think about it, look at the wires that, if you're still using wires connecting the parts of your computer, if you cut the wire and you look at it, they're really almost microscopic pieces of copper. And look at a printed circuit, and look at an integrated circuit. You do get into basically microscopic conductors. So now you're into the realm of the way your brain is wired, and I mean wired. So some of the circuits stimulate things. They may stimulate mood, stimulate actions, depending on what part of the brain it is. And in this case, the case of glycine is known as an inhibitory neurotransmitter. So what does that mean, and what is a neurotransmitter? I still haven't defined that yet. Well, where the wires make connections, they're not hardwired. They are actually the signal, which is called an action potential, is propagated along the surface of the neuron, along the surface of this so-called axon, let's say, which connects that neuron in your spinal cord to the little toe you want to wiggle, right? Right. So you have that. When it gets to the little toe, or when the neuron that connects from your spinal cord, relaying the signal from the brain to the neuron that makes the little toe wiggle, it's connected through what's called a synapse. And many of you probably heard of a synapse, the connection between. Well, a synapse is not a hardwired connection, so to speak. What happens is it is chemically transmitted. And that chemical that transmits the action potential has to flow across a very narrow gulf. It has to take a ferry across this so-called synapse from the presynaptic membrane, the cell that's sending the signal, to the cell that's receiving the signal, the postsynaptic membrane. And that molecule is called a neurotransmitter. There are particular neurotransmitters that work on different kinds of nerves and muscles. For example, to make your little toe wiggle, the neurotransmitter is called acetylcholine. So there are different ones. In the brain, there are different ones. There are GABAergic, gamma-aminobutyric acid, and different, stimulatory ones. And there are inhibitory pathways. The principal one of the inhibitory pathways being glycinergic neurons. And glycine is an inhibitory neurotransmitter, so that when glycine is secreted from the presynaptic end to get to the postsynaptic end, right, to cross that little gulf in that little ferry boat, right, it is actually, its role is to suppress or to dampen that signal. So it's inhibitory. Because that is a very relatively slow process and a complex process to get from the presynaptic cell to the postsynaptic cell, it is very prone to being manipulated through drugs. So this is where a lot of drug targeting research takes place. effect at the synapse. So let me take one where everybody's probably familiar with the initials and maybe even what they mean. You've heard of the antidepressants called SSRIs, right? What's an SSRI? It is a selective serotonin reuptake inhibitor. Okay, what does that mean? Well, it's selective, which means it's a drug that's for a specific kind of a neuron working in a particular place. Serotonin is one of the neurotransmitters. Usually they're small molecules like amino acids, like glycine, or derivatives of amino acids like acetylcholine or serotonin. So what happens is when the signal gets to the end, to the presynaptic end, to the so-called terminal button, there is stored there some of that neurotransmitter, and that impulse makes that neurotransmitter get released in little packets. So they sail across that little gulf to the postsynaptic terminal. It happens in milliseconds, of course, because it's very small distances. It diffuses over to the other side and connects the impulse, and then the action potential, the impulse, is propagated in the postsynaptic cells. And that's how the nerve impulses happen. Well, so when the impulse goes through to release the neurotransmitter from the presynaptic end, it is released now. But it all happens so fast that as soon as it's released, the cell quickly is taking it back up. There is a reuptake. And reuptaking it is a way of dampening that signal. So let's say it's been in serotonin has to do with a lot of the up mood regulation, right? Positive stimulation in the brain, in the central nervous system, and certain various circuits. So what they want to do is, well, there's not enough serotonin, so we want to make whatever serotonin there is act longer, linger longer at the synapse, so that you have a positive signal that is extended and amplified. So how do you amplify a positive signal? So they have drugs, the major class of antidepressants that we have now, from Prozac on down. What are they? They are SSRIs, selective serotonin reuptake inhibitors. They stop that cell that secreted the serotonin from taking it back up again, so it doesn't take it back up again so quickly, so the amount of serotonin signal is amplified. So that's one of the strategies. That's the main strategy in antidepressive medication. Very simplistic, but on that level it works. And of course, we think we know, it works as an antidepressant. So now, as scientists, we think we know everything about depression. It's just...
Well, I was on SSRIs.
But it works that way. That neural circuits, they work that way. I'm sorry, you were going to say.
Yeah, no, thank you for that explanation. And I was on SSRIs as a teen, and I can say, I know they're life-saving for some people. My experience was not a good one. So they don't work. And like you're saying, there's so many new emerging findings around depression and maybe different subtypes of depression. And something, you sent me an article about precision psychiatry.
Right. So, but I don't even remember that article. I've read so many recently, but I will say that this is a good intro to the topic you wanted to introduce and about the article that you sent me about the GLYT1, GLYT channels. These are glycine channels. So we have glycine receptors. There is that classical glycine receptor, which is an inhibitory neurotransmitter in the central nervous system. There is also the so-called NMDA receptors, for which glycine is a co-activator. So glycine is needed in addition to D-serine, another amino acid derivative. The two of them together activate this other sort of positive stimulating type of neuronal connection. And so lately research has been working at the synapse where glycine works. Why? Because they have had some success in major depressive disorders, schizophrenia and bipolar disorder, with supplementing with glycine. It seems that glycine has been a good thing. So what did they find? They find there's a transporter called GlyT1 and GlyT2, and one of them is for the NMDA receptor, one of them is for the classical glycine receptor. What is GlyT1? So they're looking for drugs that will inhibit the function of GlyT1. What's that? Guess what? It's another SSRI, but it's not selective serotonin reuptake inhibitor. It's really an SGRI. Selective glycine reuptake inhibitor. That's what these drugs targeting so-called GlyT1 are. They're SGRIs. And they're designed to inhibit that reuptake of glycine by the presynaptic cell so that glycine lingers more in the synaptic environment there, the microenvironment, and the signal that is propagated by glycine, either inhibitory or stimulatory, is amplified. So that's kind of how that works, and that's a new realm of pharmacological research, which hopefully will produce even newer and better drugs. Because these days, in so many years, Thorazine, that story I told, it was in my lifetime, but still ages ago. And many, many generations and iterations of antipsychotic medications have taken place, and antidepressant medications have taken place. And now there's such an armamentarium of different ones that all act in different ways, interact in different ways in different people, that really these days the art of the psychiatrist generally is an orchestrator of chemical therapies, of pharmacological therapies, a mix of drugs that will actually make the brain work in a normal or near normal fashion. That's basically what modern psychiatry has become.
Yes, I love this. Okay, so just so that everybody understands, there's this classic inhibitory nature of glycine. So what would that be used for? Like, tell me what that would cause people. How would people know that glycine was acting as an inhibitory neurotransmitter for them?
Well, it's always acting as an inhibitory neurotransmitter and as an activating neurotransmitter. It's always doing both. And my argument about the need for glycine supplementation generally, about the existence of a glycine deficiency that's almost universal in our population, is that there's an imbalance. And certainly for some people, there are all kinds of effects, genetic effects, environmental effects, psychological stress, chemical stress, everything like that happening. For some people, just providing enough glycine around will enable the balance to be correct, so that there's enough in the inhibitory and the stimulatory pathways so that the brain functions optimally. And certainly, there's something to be said for trying that first, is saying, well, if you have a problem that may be somehow related to glycine and its action in your brain, a good way to start would be to say, how about having an adequate amount of glycine in your blood for your brain to start with?
Yes.
And that's all that I advocate, actually, in glycine supplementation. I'm not talking about megadosing anything. I'm just saying, let's put back the glycine in the diet that we don't eat because we throw away the bones and the connective tissues, and don't throw them in the soup like our grandma used to do. And, instead we throw them in the trash. And put those back in the diet. So get 8 grams a day of additional glycine, adding to the 2 or 3 that you probably get from your regular diet. Take another 8 grams a day and see what that does. But the pharmacologic part, which I have nothing to do with. I mean, I'm not a psychiatrist. I'm not a doctor. I'm not a nurse. I'm not a clinician of any kind. I'm a scientist. So I just kind of figure out what's going on, and then, you clinicians can deal with it. I'm sure there's some of you out there.
For sure.
This is the basic science behind it that I'm after, right? So it may work in different ways. And one of the pharmacologic ways they're talking about it is to target drugs for this glycine reuptake, this glycine 1 transporter, GlyT1, glycine transporter 1. And I think that's the regular inhibitory one.
Yeah. Okay. And so when it comes to the NMDA receptor, because that's related to the NMDA receptor because it's a coagonist, right?
Glycine is, yes, it's a coagonist, and that is the one that recent research shows is probably involved in sleep. I used to think, glycine, a lot of people recommend it for sleep. It helps people sleep. It's been marketed that way for decades, I think, actually. And I assume it makes sense because it's an inhibitory neurotransmitter. Now, it should be noted that hormones, drugs, nutrients, receptors, and pathways for receptor activation are complex, in that they're different actors working through different kinds of cells and different kinds of pathways, but they complement each other. So if glycine is basically going to be a calm things down kind of a guy, if that's glycine, then it's going to react in different pathways in different ways, but they're all going to be sort of geared to the same calm things down. So that will lead us to yet the latest way they've discovered, the new glycine receptor, which I'll get to in a minute, or a couple of minutes. But in any case, the NMDA receptor 1, that seems to be the key one involving sleep. Not all the evidence is in, but it looks like they have identified the major pathway of glycine's action is through that NMDA receptor in regard to sleep. How? Usually these are done through animal experiments. They'll do it with rats and mice. What happens is that glycine is needed in order to enable the animal to sleep. How can they figure that out? Well, they can develop a rat which has a gene knockout, which doesn't have a gene for using this particular pathway, or they can starve it for glycine, and show that the animal has trouble sleeping. And the details of how it's allowed to go to sleep have to do with the NMDA receptor pathway, which acts by one of the ways it acts is to dilate peripheral capillaries in your circulatory system. What does that do? It lowers the body temperature. What does that do? It allows you to sleep. You have lower body temperature, lower action, lower general metabolism. So this sort of positive pathway is involved, this NMDA pathway is involved in the relaxation response. I'm not talking about the book that somebody wrote a few years ago called The Relaxation Response. I'm not sure how that relates at all. But the relaxation response of the body in terms of glycine as a necessary nutrient and the enabling of sleep, to the extent that it works that way, and many people report benefits that way, and many of my own customers do report that sweetamine helps them sleep. It doesn't make you sleepy. It's not a sleep inducer, but it does those things that allow the body to relax and go to sleep. So that's complementary to its action as a let's make the mind less busy inhibitory receptor in terms of glycine. So that works. So those work in a complementary fashion for brain function. Now we get to, so we've talked about the classical glycine receptor, the inhibitory receptor, the NMDA receptor, which seems to act mostly in glial cells, which are cells that interact with neurons rather than the neurons themselves. The neurons usually seems to be the classical glycine receptor in the brain. Now we have, we talked about the transporters, which are those channels which allow the glycine to be taken back up again where it is secreted to inhibit the signal so that the next signal can come through. And now we have another kind of glycine receptor, which is called, I don't think they've named the receptor yet, but it is...
Is it like GR158 or something?
What is it called?
I think it's like GR or GD158.
Yeah, I don't remember I don't remember these names and numbers so much, but what it was an orphan receptor. Now I could get to the idea of orphan receptors.
It's GPR158, G protein receptor 158.
158, okay. Because so when they discovered hormone receptors, not only hormones, but nutrient receptors, like your taste buds, right, which you have on your tongue and in your stomach, believe it or not. You have these act to what are called G proteins, which means the receptor is on the cell surface. And when it binds that ligand, whether it's glycine or whether it's sugar or whether it's some other thing that you can taste or that will fire up that neuron that says, give me more of this, or whatever it is. When that receptor interacts with the cell surface, at the cell surface, it sort of causes like a wiggle in something called a G protein, which starts a biochemical cascade of signaling events that initiate some kind of change in molecular behavior of the cell. So that cell then does something or makes something which changes the behavior of the organ, makes it secrete something else, and so forth. So G protein receptors very, very common. Well, when they discovered these G protein receptors, probably maybe the late 70s, 80s, I'm thinking, the 1970s, when they did, they said, okay, so this is a protein. So they isolated G proteins. They figured out the sequence of it, and then they measured systematically all the proteins that you find in the human body and human cells. And they say, well, that's interesting. We know that there's, I don't know, let's say, I don't know the number, but let's say we found there's 120 G protein receptors for different things, for sugar, for this hormone, for that hormone, for glycine, for, and so forth. So many G protein receptors, but you know what? We found in our molecular mix that there's a whole bunch of receptors, a whole bunch of molecules that we found in the mix of molecules that your living cells made that are obviously G protein receptors. Because that's what they look like. There's that amino acid sequence. It's obviously biochemically a G protein receptor, but we don't know what it's a receptor of.
Yeah.
So what do they call them? Orphan receptors. So one of those orphan receptors, the number 178 or something?
158, yeah.
G protein receptor 158 which was an orphan receptor. Now they just found out it's a receptor for glycine. It's not a classical glycine receptor, because a classical glycine receptor is one that is called an ionotropic receptor. An ionotropic receptor is basically a channel that when glycine is there in adequate quantity, it opens up the channel to allow chloride ions to come into the cell, which is inhibitory for the cell's action. That's how it works in the immune system to inhibit inflammation. It serves as like a trigger lock to hyperpolarize the membrane so that the switch, which would flip the cell on to cause inflammation, stays off. So it's ionotropic. It's an ion channel. It allows ions to come in or come out. In this case, the classical glycine receptor is a glycine-gated chloride channel, which allows chloride ions to come in from outside the cell so that the voltage of the cell does not deteriorate and the cell does not inappropriately get activated to cause inflammation, which is what happens when you don't have enough glycine. And that's most of the time these days. That's why we have all these inflammation-based diseases. So that's been my classical argument about the classic receptor. There are lots of other things, which is what you wanted to talk about today. And the other one is that new receptor, which is a G protein receptor, which makes it a metabotropic receptor, because that G protein now enables a bunch of biochemical reactions to commence. So it starts a cascade of metabolic events, chemical reactions within the cell that changes the signaling and the behavior of the cell. So it's metabotropic, and as old G protein receptors are metabotropic, and that's the new glycine receptor. Right now, we can just call it the metabotropic glycine receptor.
I love that. Can I take a second to say, anyone who's not a biochemist, this might seem confusing, but in biology, in biochemistry, they want to kind of define the type of receptor, which is exactly what he's doing.
How does it work? Yeah.
Yeah, metabotropic, right? And all of these different receptors, they do different kinds of things. They're essentially like sitting on the outsides of cells, waiting for different substances to come connect. And then when those substances connect, different things happen, whether it's to metabolism, letting ions in. And so all we've talked about so far are the kinds of receptors that are attaching to glycine, that glycine seems to be really important for.
Right. So the first two, the classical one and the NMDA receptor, for which it's a co-activator, those are ionotropic. And so the metabotropic receptor is a new one. And there is yet another one that I've talked about that's not even called a receptor yet. It's a molecule on the surface that has to do with the end stage of inflammation. That's called ningerin-1.
And this is our fourth, because
so that's another one, yeah. So these are all forms of glycine receptors. And ningerin-1, again, complementary function, because the initial function of the classical glycine receptor in the immune system is to stop the initiation of inflammation, and the ningerin-1 happens at the very, very, very last step of inflammation to stop inflammation at the very end, so you don't go and die of septic shock, for example.
Yeah, and if I remember from your book correctly, it's like adequate glycine will inhibit that last step, because the ningerin-1 won't kind of aggregate and lead to that explosion, or is that correct?
Yeah, ningerin-1 is a different kind of receptor entirely. That's why they don't even call it a receptor. It is because it's a specific substance that binds glycine specifically. It will bind other things to some extent. Taurine, for example, will substitute for glycine in many situations to some extent. But it specifically binds glycine, and what it does is it seems to have a physical effect, actually, of keeping the cell from exploding.
Yes, right?
It stabilizes the cell membrane of a cell that's ready to go, ready to explode. And when do you need the explosion, the final step of inflammation? Well, you get bad bugs like anthrax and so forth that need to have really a lot of severe inflammation in order to stop the infection before the infection kills you. You need to have this sort of nuclear detonation at the end. And when this is inappropriately initiated, like, for example, by COVID, spike protein, that initiates the so-called cytokine storm. And ordinarily, you can withstand the cytokine storm. It is sort of self-limiting, so you may survive. But many people died. Who died? Well, people who were overweight, people who were diabetic, people who had cancer, people who were at advanced age, all kinds of people who have lower levels of glycine than the norm. So glycine is not enough to stabilize the, and then you wonder, well, how is glycine going to stabilize the reaction? If that inflammation has now gone way past the classical glycine receptor, how is any kind of reaction, especially if that final step happens? Now you've got the cells literally exploding, spraying inflammatory compounds all over the place. How is such a reaction ever going to be self-limiting? Well, the body is very wisely designed. How that happened is another question, but the body is very wisely designed by whatever means, so that it is self-limiting because the concentration of glycine is much higher inside cells than outside cells. In fact, cells of the body generally pump in glycine. So even though the glycine is not high enough in the blood or in the lymph, in the plasma, all the fluid surrounding the cells of the body, it's even higher inside the cells. So when it's deficient, it's very deficient inside the cells. So what happens is in a normal, let's say the normal range of glycine, or maybe high normal or a little higher than normal, maybe a really healthy range, even if the immune system is appropriately stimulated to cause inflammation, and that inflammation goes to the very last step, once those cells explode, spraying inflammatory compounds all over the place, it's also spraying more glycine all over the place. Every time a cell explodes, the concentration of glycine in the nearby area increases. So it's self-limiting because what it does is it stops other cells from exploding, and it stops the initiation of the inflammatory response in neighboring cells, depending on how far away they are. So you can see how the cytokine storm can be limited. But in fact, if you have enough glycine, you don't get the cytokine storm in inappropriate circumstances. In COVID, it doesn't do you any good. Your immune system's trying to kill this virus. Well, guess what? Viruses aren't alive. You can't kill them anyway, right? Antibodies act through other means, killing infected cells and so on, but this is not going to help. So, for example, I and various friends and family and customers of mine, we went through the pandemic, everybody got COVID, right? But nobody got a serious case of COVID. I got a case that I would say was a moderate case. I had to get about 12 hours a day of sleep. I was kind of out of commission, just, around the house, not doing much, but I was not bedridden. Certainly didn't need to be hospitalized or anything, but I felt awful. I thought that COVID was a really, really rotten intestinal flu kind of disease. I got diarrhea for a day. I spiked a fever for a day. I lost my sense of smell for a couple of weeks. I came back. My taste was different. All of that stuff. So it was not fun at all. But guess what? No coughing, no sneezing, no wheezing. No shortness of breath, nothing. I even had a friend and customer who got through COVID just fine. And she had just the prior year, she had just recovered, and it took her 10 months to recover from lung surgery. Her lungs had already been compromised with air bubbles and so on and so forth. And, all you need is COVID when your lungs are already compromised, right? Well, it didn't affect her lungs. She was fine.
Wow.
And so you put it all together, and I know there are other people who had, whether COVID or long COVID, had another friend whose dad was about 70, I guess early 70s, and he got COVID, and he was admitted to the hospital, and they put him on a ventilator, and he was still alive, but barely alive, and finally they said they had to take him off the ventilator. My friend had to beg him, please keep my dad on the ventilator a little while longer, a little while. I guess they finally gave him enough steroids, right, anti-inflammatories. That's now the standard protocol in COVID. And he got those, and they said, well, okay, so we'll release him. So he said, well, so they released him, and my dad came home, but he's completely non-functional. He's just like a dead man in bed, like constantly. You can't wake him up. He can't get up, can't do anything. So maybe your sweetamine will help, my glycine supplement. So I said, sure. My wife was very careful. She said, we can bring him some sweetamine. Drop it off at the front door, This is COVID, remember, all with the mask.
Oh, yeah. How do I forget?
Ridiculous nonsense. So anyway, so we dropped it off at the front door, and he took it in, and he made his dad some tea. And then I get a call, about a week later, I guess, and I said, how's your dad doing? He said, it's like a miracle. He said, you're like an angel who came in the night, dropping off your wonderful product, the sweetamine. He said, I gave Dad a cup of that tea, at night. The next morning he got up and he went about his business. Like he was over it, not sick anymore, because he was so glycine deficient. Because again, your body makes less glycine as you get older. So there's age, there's diabetes, all kinds of other symptoms. So who was it who died from COVID? The very elderly. The very elderly, the most
and the
likely
metabolically unhealthy.
Yes, the most metabolically unhealthy, but not just, that's kind of general, right? Metabolically unhealthy. What does that mean? We're talking about glycine here because glycine, and that's where the science comes in. My old friend Occam's razor, that scientific principle that most scientists seem to be unaware of, that when you find that something works, and you find that active principle and that active principle works, alone without the whole mixture. Chicken soup is good. Chicken soup is great. And there are all kinds of wonderful things in bone broth and chicken soup. But in terms of inflammation, it's the glycine. How do you know? Because if you just use glycine, the glycine does the trick, and that's the simplest answer. And Occam's razor, the scientific principle, says the simplest explanation is going to be the right one, almost certainly. And so you don't reject. People say, "Oh, just glycine," and these days people think the opposite. They say, "Oh, just glycine can do all that one little thing." Oh, yeah, sure, glycine's the end of the world. Oh, here comes Dr. Glycine, here he goes again, it's all about glycine and this and that. Occam's razor, you don't reject a hypothesis because it's too simple. You reject a hypothesis because it's too complicated. Yeah. And if the simple one works, it's the answer. As I liked it, my favorite example is, that's why we understand that the Earth and the Sun that the sun and the planets do not revolve around the Earth in complex epicycles. Rather, the planets, including the Earth, revolve around the sun in simple elliptical orbits. That's the simplest model. We reject the complicated one, and we go with the simple one. And guess what? That's the one that works. Yeah. That's the one that's correct. The Copernican model of planetary dynamics. So science is science. Intergalactic or microscopic or subcellular or whatever level you're working at. Scientific principles are scientific principles.
Yeah, and I love that because if we are, 8 to 10 grams per day, that's a big amount, deficient in glycine, something that our body needs that we are not getting because we're overweight, not eating connective tissue or bones anymore, right? And certain things deplete glycine. So it makes sense that instead of going to these other hypotheses, we simply fulfill our nutritional need. And I want to tie this into, right, you're saying so inappropriate inflammation can happen when we don't have not even adequate amounts of glycine, not like you said, between 150 and 350 micromolar, but like 500 plus. Yeah, so that can dampen inappropriate inflammation. Now, for some people with depression, they've realized a subset, 30%, seem to have inflammatory markers that are elevated. Yeah. Specifically like CRP.
So that's the latest. That's the latest research, right? Is about how we don't know what the connection is, but it seems that 30% of the people with depression also have chronic inflammation.
Talk about that. Is that microglia in the brain?
So let's go back to our friend Occam and his razor and the simplest explanation. We say, if it's inflammation. So inflammation is what's causing the depression, right? If we have depression and we have inflammation, then one thing, A is causing B. Well, maybe not. Maybe it's something called C, which is causing both A and B. What we're looking at are symptoms of a common cause rather than some kind of sequential actors. They are both reasonable hypotheses. Yeah. So, but which one fits the data? Well, if you know enough about glycine, and probably the researchers who published that paper they don't usually talk about glycine or know about it or understand it, that if they knew that inflammation is largely due to glycine deficiency, and if they also knew the known facts about how glycine works in the brain, they would probably instantly reject the sequential hypothesis that something, we don't know what, causes inflammation, and in some way, we don't know how, the inflammation causes you to be depressed. That's kind of a very vague and nebulous and not very useful hypothesis. But if we say, we know that inappropriate inflammation is caused by glycine deficiency, and we know that glycine has these functions in the brain, which has been found useful in helping with conditions of major depression, for example, in clinical trials, it looks like both inflammation and depression are two effects of a common cause. And we even know it's a common receptor, right? It's the same receptor in the immune system that causes inflammation. That's the inhibitory receptor in the central nervous system for glycine. So the answer is kind of right there. And that's sort of vague, nebulous. Well, maybe it's causing this and this is causing that. You don't need Rube Goldberg was probably, I don't know if he was a very good scientist. I don't think he was a scientist. Rube Goldberg, the guy who one thing would do this to that, and that would trip another lever and another lever and another lever. And we find that there are many biochemical signaling pathways that do just that. All of them are like kind of Rube Goldberg, this activating that and that inhibiting this, so it doesn't activate something else. And we do have pathways that work that way. But to assume that kind of complexity going in, not a good idea when we're looking for that connection between inflammation and depression, because we have certain known facts which link them very clearly through the same receptor. So what are we talking about with all this other hypothesizing? It's unnecessary hypotheses. That's what Occam's razor says. You reject unnecessary hypotheses. So the hypothesis that somehow inflammation through some mechanism is causing depression is an unnecessary hypothesis. They're just both symptoms of the same cause.
So your hypothesis then would be that this 30%, this subset of people who are depressed, might just be glycine deficient.
Well, they're probably 100% of them are, and including 100% of people who aren't depressed. But glycine deficiency manifests in a lot of different ways. And some people may just have excess or inappropriate inflammation. Some of them may just manifest depression. But it may very well be that even the ones that aren't suffering obviously from some sort of systemic inflammation are glycine deficient, and you may find that a lot more than 30% of the depressed ones are suffering from glycine deficiency. So since we know that most everybody's deficient in glycine, why not just correct the deficiency, correct the known deficiency, and then see what we got?
That's what I was thinking. And with the Orphan receptor research, I haven't seen any depression trials where they've given people glycine or even these inhibitors. Have you seen that?
Years ago, there were a couple. That's when I was first formulating my product. I remember before it was a product, it was just self-experimentation. I would say, well, you know what? I think I should take at least 10 grams a day of glycine myself and see what happens. If all my theoretical suppositions and propositions are correct, the least I can do is do a self-experiment with something as harmless as glycine. So let me just take 10 grams a day. Well, how do you take 10 grams a day? I mean, it's sort of like sugar because it's sweet, but it has kind of a bad aftertaste. It's not a pleasant sweetness. Right. It's got that sweetness, but it's got a bad aftertaste. That's why I formulated it with, some other material. And I have a little bit of erythritol, not a lot, That's something that's not appropriate as a bulk sweetener, but I don't use it as a bulk sweetener. But it's a little bit of a natural sweetener. I put in some taurine originally because I was selling it as a drink, and taurine was not only something good to eat and something which complements the effects of glycine, and it's a good supplement, but taurine is also a tasteless acid. And when I was formulating a drink, in order to formulate a drink to sell, it has to have a pH of below 4.1 to be shelf stable. Otherwise, botulism can grow in it.
Yeah.
So I was looking for something to modify the taste, and originally I came up with stevia and proline, which is another amino acid that also complements glycine. It's also rich in collagen, but also a little bit of stevia. But I also then needed to reduce the pH. So after I had the formulation that tasted the way I wanted it to taste, I had to acidify it. So I put in a little vitamin C, but vitamin C is also sour, ascorbic acid. So I needed to get the pH lower, but not make it sour. Taurine lowered the pH as much as I wanted, but without contributing to the taste. So it may contribute to the taste of the glycine, but itself it's tasteless anyway. So I ended up keeping that in the formulation. I thought that was a very good addition. But that's how I did that, is I wondered about how do I take 10 grams a day? And so what I ended up making, I started making a glycine jello kind of formulation. I started with jello because jello, a standard serving, a 4-ounce serving of jello has about 500 milligrams of glycine in it as the gelatin, which is really denatured collagen. So I thought, let me start with something with a lot of glycine in it. And so I developed it so that I would have a portion of jello. And I used unsweetened gelatin. And then I didn't want to add any sugar or use sweetened gelatin, so I took unsweetened gelatin. You can add like your favorite juice to make your own jello, if you right. Jello is copyrighted, but you can make your unsweetened, or you can make your own version of that by taking unsweetened gelatin and mixing your own fruit juice with it. So I started with sour cherry juice. So I figured between sour cherry juice and 8.5 grams, 9.5 grams of glycine per serving, and the gelatin to make it 10 grams, I had a way to pleasantly give myself 10 grams a day of glycine. In fact, for a month before I started selling this to anybody, I took 40 grams a day. I had 4 servings of 10 gram servings of glycine, so that I knew that it was perfectly safe and tolerable and everything like that. At least for me. I mean, I'm sort of a Joe average that way metabolically. I don't have any kind of genetic variant that would make me kind of out of the ordinary. So what's good for me is not necessarily good for everybody, but it's kind of a Joe average, which I'm sort of Joe average, that way. And so that's worked out well because most people like it. And if you don't like it, you can get your glycine from Paleo Valley. Again, get bone broth. Just have it all in bone broth. If you take 4 times as much glycine or 4 times as much collagen as in bone broth or collagen supplement or gelatin supplement. So instead of 8 grams a day, you take 32 grams a day roughly. You'll get all the glycine you need. But most people don't like to have that much bone broth every day. You need to drink about a quart of it. So it's, have a little this way, a little that way, that's fine. Just get your 8 grams a day or so and get your glycine to be a normal, healthy level. And then see if that helps with your mood or with your sleep. And so people
Did it
a lot
help
of times they say,
with your mood or sleep?
Me?
Yeah.
I never had any trouble sleeping.
Or mood? You didn't have mood?
I could be giving a lecture on YouTube at full, put myself to sleep, I mean, I don't have...
Okay, have you heard from customers
what was that?
Have you heard from customers that there's like a primarily antidepressant or even anti-anxiety effect, or have you...
No, not really, no. But sleep, yes. Sleep, many of them said sleep, but it doesn't make you sleepy, so it's no problem taking it in the morning. That's what I take it first thing in the morning.
For some people, and that's one question I wanted to go over, so for most people, you will not feel sleepy. I've even seen glycine supplementation improve mental alertness the day after, but for some people, it does. There's a paradoxical response. Some people are like, whoa, I'm so tired, or whoa, I feel anxious. What's happening there?
That you find true with anything that acts in the central nervous system. Glycine is a neurotransmitter, and when you take it as the free amino acid, it's getting absorbed very rapidly. So I would say for people who are having untoward effects like that, and there are some, as you say, a small minority, but there are some. I say then get all your glycine through something like collagen or gelatin or bone broth. Get your glycine that way, because in that way the glycine is there as an amino acid part of the protein molecule, the collagen protein molecule, or the fragment of protein that is in that formulation. A collagen hydrolysate, let's say. And that way it won't get absorbed until you digest it. So that way it's absorbed. It's all absorbed very nicely, but it's absorbed more slowly. So if you are that way where you're reacting, either hyper or hypo or one way or the other, if it's having these rapid nervous system kinds of effects on you, then, or on the GI tract, another small minority also have GI upset from it. Few people get diarrhea from it. Again, these are very rare. Most people, you don't notice anything as far as those things go, but it will, except for aches and pains and anything that's due to inflammation. But for those people, I would say don't use Sweetamine or any other free glycine supplement. Use a supplement that's like Paleo Valley's bone broth. Delicious. So you found a way to get glycine deliciously. Yeah. And so did I. So take your pick.
Yeah, exactly. And right? Now, so for
different strokes for different folks.
That's exactly right. And we're just trying to provide as many different ways to fill that glycine gap, because we both believe that it's probably one of the most important things you can do in modern nutrition.
Absolutely. Yeah. When it comes to most of the illnesses that make people sick and die these days, in just about any way, it's not a drug. Not a drug. I don't sell it as a drug. I'm not a clinician. It's food. It's a nutrient that we don't get enough of.
Exactly. And I wanted to cover a few other conditions really quickly, and that's schizophrenia and OCD. So you got these papers that I shared papers where they're adding large doses of glycine to treatment for schizophrenia, for example, like 40, 50, 60-gram doses, and finding a reduction in negative symptoms. You've also told me you didn't feel like those results were super significant.
Well, I had read those. There were some papers to that effect some years ago.
Yeah.
When I first started, formulating glycine and testing it out and marketing it and doing the basic research, then that's exactly how I started on the journey of trying to find a palatable or an enjoyable way to take your glycine as the free amino acid, because it started with a sweet taste, and I figured, well, it should be a way to, get this to work. And I think I was very successful with it. And I think most of my customers agree. But that was a long time ago. So that was research that was done way back when. And I don't know that I really looked at the recent one. I just saw the abstract about how they're trying it with 50 or 60 grams a day. And in those days there were. And I marveled at how they would say, how would they do this 60 milligrams a day? They would take 20 milligrams, not 60 grams. They would take sort of 15 grams at a time and put it like a quarter teaspoonful under the tongue. Put this under your tongue, because they were thinking like doctors, like taking drugs, taking medication. Okay, glycine is very rapidly absorbed even before it gets into the digestive tract. Just under the tongue, it can go right through sublingual, like B12, other things are sublingual lozenges. So they were administering it sublingual, and I also remember not only they were barely significant results that didn't really matter too much clinically, but also there was a very, very tough time with compliance. You had people who had severe depressive behavior and brain function to begin with, and now you're giving them something that's really unpleasant to do. And it was very difficult to get them to comply with the protocols. So now I do have to revisit. It may be that it's more effective than the earlier research seemed to indicate, and maybe just the way to do it is to make sure that you're administering the glycine in a palatable form.
Yeah, I think that would change it, because that's what I've seen in the trials too, is compliance has been difficult, but there were significant changes in their negative symptomology, and they tied that to the NMDA receptor and maybe the ability, to get their brains moving or...
Right, because that would be the more the stimulatory way. So a lot of these drugs, antipsychotic drugs, for example, they are stimulatory, in some ways. They're stimulatory in some ways, inhibitory in other ways.
Yeah.
It's like LSD is a stimulatory drug, which causes psychosis.
Yep.
So that's where the antipsychotics actually go against those dopamine receptors. Right, dopamine is the big yippee neurotransmitter everybody talks about. Oh, I want to get my dopamine reward. Well, that's what LSD actually takes the dopamine, and LSD is basically a dopamine receptor agonist. Right, it is a neurotransmitter that acts like dopamine, except when it binds to the dopamine receptor on the postsynaptic neuron, it doesn't let go.
Oh,
so it just keeps it going, and it just drives you crazy. Literally, that drives you crazy. That is how LSD literally drives you crazy, induces a state of toxic psychosis. So, this is how. So there are drugs that you try to do to agonists and antagonists. You want to increase the effect, decrease the effect, and of course they try it in vitro systems. With the chemicals, with the neurotransmitters, they check out the binding, and then they'll do it in animal studies and see if it works in the animals and doesn't hurt the animals, and all of these different hurdles that these drugs have to get through before they can be tested in people, let alone approved by the FDA or whatever authority is operative in whatever country you are in.
And then the other thing is OCD. There was 2 trials that I'm aware of. One's a case report where I've mentioned this before, but significant changes with 60 grams a day.
Yes, I saw that one, yeah.
Yeah, so that was crazy.
It was a case report, it just made it go away.
Yeah, made it go away. His very severe body dysmorphia and OCD symptomology that kept him housebound, essentially. And then later was able to go back to school, improve,
But that was with glycine, right? That was
just
with
glycine, 60 grams. And then there was an OCD trial too, where the compliance issue, I believe it was around 40 grams a day. A lot of people dropped out, but the people who did remain ended up seeing good results, and some of them continued to take glycine after the trial.
Well, did they also give glycine in that trial with the sublingual deposit or something like that?
I don't know. Well, no, or it was something like that. And I remember reading that people kept dropping out, and I thought, oh, sorry, we just got to figure out. My question on those trials with the 50 and the 60 gram doses, do you think there's a case where, you recommend about 10 grams a day?
Right.
Do you think for brain-based issues, there might be a reason why they need huge mega doses?
Could be. Well, it could be. I mean, it's entirely plausible that it could be used pharmacologically as a drug, using supernormal amounts. So, and I don't know, but again, I'm not a clinician, I would be interested in the literature, but I wouldn't say, hey, this study says that they gave 50 grams a day to somebody, so you should try it.
No.
I'm not going to say that. Check if you want to use glycine medicinally, because even at the dosage of 10 milligrams a day, a normal amount, you have some condition and you're taking some drugs for it, some medication, and it may be interact. I get questions sometimes, will glycine interact with my medication for this or that? And I say, if you're under the treatment of a doctor of any kind, then want to know if glycine is going to affect the treatment or affect the medication or anything like that. 3 magic words, ask your doctor.
That's right.
And hopefully you have a good one who will give you a thoughtful answer.
I just wish they could do some with smaller doses and see, because they're just like something people could do, like with sweetamine or something. But, you can. You can, anything that's safe, you can put as much as you want. It's like, for example, this whole story with erythritol. Oh, erythritol, you have erythritol in your product. As I say, I put a little erythritol in sweetamine. It's got 800 milligrams of erythritol in a serving of sweetamine. The research that implicated its ability to increase clotability and perhaps increase the frequency of major adverse cardiac events was 37 times as much. 30 grams, 30,000 milligrams instead of 800 milligrams. You take 37 and a half times as much as you usually eat of any nutrient molecule, and it will probably make you sick or even kill you. But erythritol is actually naturally in your body. Your body naturally makes a little, and it's probably better for you than sugar anyway. It is a sugar substitute. And it probably, even at those concentrations, as a bulk sweetener does less harm than sugar. But I'm not talking about using it in those terms. So I wouldn't recommend erythritol or anything like that to be used in, multi-gram quantities. Milligram quantities, well, it's normally there. Your body normally makes milligram quantities of it. So it's not going to hurt you. And that's why it was originally designated, generally recognized as safe. The FDA says erythritol is safe to take up to 80 grams a day. Wow. That's a lot of erythritol.
That's a lot. And that is, so what is that? That's 100 times what I put into a daily serving of sweetening.
Yeah,
don't take 100 packets of sweetening every day. Please don't.
I love that you can.
I don't think it's going to kill you, because you can't. It's almost impossible, if not impossible, to kill yourself just taking glycine orally, because that's really, really, really safe. But it can induce some short-term discomfort, and if it does that at the ordinary amount that I recommend, 8 grams a day, 10 grams a day, then don't take it as the free amino acid, as I said before. Take it as in the protein form, like collagen or gelatin or bone broth, whatever.
Yeah, and we have lots of videos on how you can eat enough glycine too. So yeah, pick your version, but just getting the glycine is
just
important.
Get enough, yeah.
Just a few more questions. So I've heard too that it can be neuroprotective in the event of stroke. I'm also wondering what you think about the NMDA story and how it applies to Parkinson's and Alzheimer's. Do you think glycine could potentially be protective?
Yeah, well, that invokes the other mechanisms that have to do with inflammation, right? Parkinson's, for example, is a deterioration of brain tissue, which may be one of those consequences of chronic inflammation. Yeah. Just as there are macrophages causing inflammation in every system of the body, every organ of the body, you have macrophages in the brain, and they are responsive to glycine. And that acts as a trigger lock there. So if you get a concussion, all these sequelae, all these problems downstream of traumatic brain injury, a concussion or something like that, in my own experience, as long as I have enough glycine, they don't happen. I mean, I had a pretty substantial concussion about 8 years ago, and I think I've told that story before. But basically, I I fainted momentarily because I was dehydrated and tired, and I had a glass of wine, and in my head, I fainted for a moment as I was getting up from a chair at the kitchen table, and hit my head, wham, right on the hard edge of a hardwood table. And when I came to, I was on the floor looking up. I was kind of dazed and confused. And my kids were there. They were asking me questions about, hey, Dad, is everything okay? How do you feel? What about this? What about that? And they would ask me questions to see if I was confused. And they said, what about Sweetamine? I said, Sweetamine, what's that? That sounds like a nice name for a product. They said, uh-oh, Dad's really gone now. The big creator of Sweetamine and everything. We better call 911. So they did. And so I was taken to the hospital and all that. And within about 2 hours, my head stopped hurting. And after that, no other sequels at all. It just healed. That was, as I said, that was over 8 years ago. I never had any kind of follow-up symptoms at all.
I love that.
It was like the classic, old Western movie, where the bad guys come and they clunk the security guard on the head with the butt of the pistol, and knock them out, So that's basically what happened to me. These days, everybody say, "Oh my God, they're going to get traumatic brain injury, chronic traumatic encephalopathy," and they probably will because they're all glycine deficient now. So, it's part of the modern condition, and I'm doing the best I can. I used to be just a scientist. I was never a businessman, and I never aspired to be a businessman. Just a teacher and a researcher. And I ended up starting this as a business because that way people can find out about it and we can talk about it on podcasts and all of that, instead of just doing it as a scientist and slugging it out in paper with all the various medical societies and journals and everything like that to try and convince them, yes, glycine really is essential. Yes, glycine deficiency explains so much of disease in the modern era. And I state these things as facts because I know them as facts. If you ask around to the mainstream scientists and doctors, they'll say, "Oh, I've never heard that. The situation is complex. It's not so simple." All of these, They get all these other end. Now, Brind's a quack. No, I'm not. I can't be a quack because I'm not a doctor and I don't claim to be.
Right. Just a researcher.
But
I love that.
Real science, and you can buy my book, The Glycine Miracle, which explains it all. That's just search The Glycine Miracle or search my name on Amazon. I don't sell it on my website because it's not food, and I have to charge sales tax, and it gets into a whole other kettle of fish.
Yeah.
So all I have on my website, sweetamine.com, is a link to the Amazon page, and you can order it that way. But I just get the book however you can, get glycine however you can. Don't worry about buying. It has to be my product or my book or anything like that. But if you're listening, and you've been listening this long, that's already a good sign. In my book, you'll try it. And when I sell it, by the way, it's $14.95 if you use the promo code Autumn, right?
Yeah.
Is that the promo?
Is it maybe Paleo Valley?
No, maybe Paleo, I don't know.
I'll put it in the description.
Well, you would know what it is.
I am not a sales girl, but
that gets you the $5 off your first purchase. And not only that, for $14.95 you can get a 12-day challenge. So you can try it for 12 days, which is more than enough time to see if the aches and pains that you're suffering from, which is why most people take it, is caused by inflammation. Because this will help. Usually in 2 or 3 days. And if it doesn't help you, or if it doesn't agree with you, just tell us. You don't have to send back any empty packages or boxes. Just tell us and you get all your money back. It's sold. That's what risk-free means.
Yeah, thank you. And
that's the way I've been selling it for over 15 years, and it's been working out, very well that way because I don't try to trick you into getting a subscription. We have subscriptions only because people want them. But if you buy a 12-day challenge or a whole box of Sweetamine from us, you're not going to get any more unless you proactively go to the website or call up us on the phone and order more. Otherwise, you won't get more. Yeah,
I really appreciate your approach for that. And I will put all those links down there. And then I wanted to say that this really matters to me, especially for your research and connecting all those dots, because I have 2 genes for Alzheimer's, and I know that my microglia and inflammation in my brain is a big thing I want to inhibit. And so that's why I wanted to share, because there's many ways it works in the brain, but there's also in that GlyT1 receptor paper, they were talking about addiction. They were talking about using it for epilepsy, Parkinson's, Alzheimer's, neuropathic pain, stroke. And so all of these conditions are seemingly related to glycine.
Yes.
And maybe just supplementing or getting enough through your diet could be helpful. So
it probably will make those things go away for at least some portion of the affected population.
Yeah. So can you give us, to wrap it up, are there any brain-based supplementation recommendations? I know you're not medical advice.
Well, I do have one, and I found it out kind of by mistake from what customers told me. Like, I get testimonials that say, thank you, Dr. Brind. I started taking your product, Sweetamine, a glycine supplement, in order to think it would help with the various, because I was suffering more and more aches and pains in my joints and everything. They were really, my back was really bothering me. He said, and then I realized after taking it, I had been getting migraines of increasing severity for some time now, and it was getting to be at least once a week. I was getting these really bad migraines. Man, I don't get migraines anymore. Well, what do you know? Well, my own daughter was getting migraines also, and they went away. But then other testimonials from other customers, some very dramatic, like, I'm a woman in my 30s, and I've had 3 kids, and I've had horrible migraines like almost daily for years and years and years, and my life's been hell. And they've gone away, So migraines is another thing. And now I had not followed the research on migraines as to what causes them, because, there were all of these, it was a mystery, take more Excedrin, and it's an Excedrin headache. No, it's a migraine headache. It's take this, take that. And they had increasingly, and my daughter, as I said, she had tried some of the stuff that was specifically for migraine. And it was like really bad on side effects. So she didn't take those on an ongoing basis. And then I realized that some of these new things that come out, like these MABs, these biologics, monoclonal antibody, biologic drugs that will work. I think some of who's the Venus and Serena, the tennis players?
Venus and Serena Williams.
Serena Williams, yeah. They've done some ads for drugs that affect that stop migraines. And how do they stop migraines? Well, it turns out, what are the MABs? The MABs are antibodies to a compound called tumor necrosis factor alpha.
Yeah.
Tumor necrosis factor alpha. What is that? Well, that is one of the chemicals that your cells secrete in response to a glycine deficiency or in response to a provocation of inflammation. It is near the top of the inflammatory cascade. So migraines, definitely another one of those, certainly a brain issue, we'd have to say, because your brain hurts.
Yeah.
Do that, right?
Absolutely.
So migraines are definitely due to inflammation. Definitely the first thing to try. It's the first thing to try for anything. Or just the first thing, if you know, just because of your own dietary patterns, that you are glycine deficient, because most people are glycine deficient, try it. That's what I did. I didn't know I was glycine deficient. I just thought, it's not going to hurt me. And I have a feeling that glycine will help, and I'm not sure how it will help. So let me try taking 10 grams a day, every day after day, month after month, year after year, from 2008. And within about 2 years, I found these dramatic results, like falling 4 feet onto a concrete floor onto my tailbone, getting a severe sunburn, conditions that should have produced, that I was certain would produce severe inflammation, and they didn't. The most dramatic experimental results I've ever had in my lifetime as a scientist. So I just had to find a way to pass that on so that people could actually take advantage of it, and starting a company and selling it. So that's what I did, and that's what I do. And and of course, that has added to not only publishing a book about it, but platforms like this podcast to educate the public about it. I don't care if you try my product or not, for my sake, enough people try it so that the business is self-sustaining. And that's good enough for me. What's good enough for you? That's what I want you to find out. When I did this, at my college, I know a lot of students tried it, and they would buy 1 box, and that's all they would buy. Why? Because you can get glycine as a bulk powder for free, just as glycine, if you want to take it, if you want to formulate it yourself. Because when you actually make a product, as you well know, packaging the product is more of the cost than the ingredient itself in many cases.
I know.
So, but that's what it takes to make it convenient, to formulate it so that it's tasty, so that it's this, so that it's that. And if you keep the price pretty much negligible, most of my customers get my product for less than $1 a day, because they buy it when I have it on sale, and you buy a 6-pack or instead of a 1 box at a time, that kind of thing. You get it for less than $1 a day. So how much is $1 a day? Oh, I can get it for 20 cents a day. Oh, okay. Well, if 80 cents a day makes a difference to you, and maybe it does, then do it the cheap way.
Yeah, I love that.
Or you can do it the easy way if it's still cheap.
Yeah.
So that's it. That's it. People say, "Oh, you charge so much." I mean, I'd say, wait a minute. Lots of people go to Starbucks and Dunkin' Donuts, and everybody knows you can make your own coffee at home for a small fraction of the price.
It's so true. Convenience and enjoyment are really big pieces of the puzzle for many people, for a lot of people. And so they're
just relative expense. If you have something that's close to zero, a million times zero is still zero.
Yeah.
10 cents a day is very cheap, but so is $1 or anything in between.
I love that.
Generally speaking.
And I want to just wrap this episode up because there's few questions from the I think I know what you're going to say to these, but I just want to ask 3 questions. But first, just some dosing recommendations. Dr. Brind, we have a glycine gap of around 8 grams a day because of what we're not eating.
On average, yeah.
On average. And that's what Sweetamine is designed to fulfill. 8 grams in one dose that you can add to food. You can also, like we said, do it with bone broth. Now, people also ask, do I have to take it with food, without food, every day? Yes, you have to take it every day. It's not something...
Yeah, but it doesn't matter when,
Doesn't matter when. You can have it with food. And are there any other dosing recommendations? I think it's pretty easy. You just
Well, sometimes if you find, we were talking about mega dosing, etc. Sometimes, the 8 grams a day is a one-size-fits-all. So sometimes if you're very glycine deficient and you have a condition that's kind of stubborn, like I've had customers who say to me, I've been plagued with plantar fasciitis for years, and I tried your product, and it seemed to help a little, but can I take more? Can I take 2 packets a day? And I said, yeah, you can take 2, won't hurt you. Well, then she got back to me and said, that did the trick, 2 packets a day. I tell that story so some people say, oh, so I should start with 2. And I said, that's great for my business.
Yeah.
But if you don't need 2 packets a day, just start with 1. And then if you need more, you can take more. If you start with 2 and it works, you're going to think you always need 2 when you may not.
Exactly. And some people are going to be highly sensitive and might want to split that up.
Yes, that's right. You can sometimes if it bothers you a little bit, whether it's the taste or some GI discomfort, you can take it with something else. Take it maybe with food more, or take it in 2 or 3 separate. My wife takes it in two tranches a day, one in the morning and one in the evening. Once with tea and once with grapefruit juice. You can take it however you like.
Okay, and the point is just fill that gap. So here's a quick question.
And if it doesn't work, just get your glycine somehow. Get your glycine every day.
Yes. So my quick questions for you. One is she's been on anxiety and depression meds for almost 4 years because she couldn't sleep. But with the meds, she sleeps only 4 to 5 hours. So, basically she's having these problems. I feel like I have a very active nervous system. Before I was diagnosed with anxiety, I was very sensitive to smells. So she's saying, I watched your videos on glycine. Is it all right for me to take this with my anxiety meds? Or is it better to just take glycine and no anxiety meds? I think this all started when...
Well, I don't think it matters, It shouldn't affect. There aren't the anxiety meds. She's probably been around for a long time, and I think the only interaction. When I said, remember before I said, well, always ask your doctor, and it's always because that's just because I can't give you medical advice, and because even if I were a doctor, I'm not your doctor.
Right.
I don't know you. I don't know, and when patients. Or when customers or people generally tell you, well, I have this and this and this, and when I did this, that happened, or I did that, this, you don't usually get a full story. It takes, if I ask you about details of any aspect of your life, even if you want to tell me everything about it, in any given encounter, you're not going to remember everything about it. You're not going to remember everything that might be important from a medicinal point of view or not. So always you have to ask your doctor. Now, it's true that sometimes the doctor will say, don't take any supplements, But all I would say is the 1 packet a day is meant to be food that everybody should be able to have every day as part of a really complete diet. People say, oh, well, I have a complete diet. If you don't have enough glycine in it, no, you don't. That's really a key discovery, is that what we think is a complete diet is not a complete diet.
Yes.
And just the essential amino acids, the classical 9 essential amino acids are not enough. Glycine is also essential.
Yes,
and so
perfect.
Have it, whatever else you have, just have that. And I think it's just there is one antipsychotic that glycine is known to lower the efficacy of. And
that's clozapine?
Clozapine, yes. So if you take clozapine, then ask your doctor, look on WebMD and see clozapine and glycine. Glycine may lower the efficacy of clozapine. That's the only one that I know of.
Okay. Second question. So people say they get anxious, agitated when they take glycine, and a lot of them in the comments are saying, well, this is because of COMT or this genetic variant.
COMT.
COMT. Do you know anything about that? Is that playing into this here, or should people be aware of that?
What, that it's increasing the activity of COMT?
Yeah, or that maybe some people with slow COMT might be differently affected when they take
because COMT, it's like when we were trying to mimic methionine deficiency. COMT, remember, it's glycine and methyltransferase is the clearance enzyme. COMT is catecholamine O methyltransferase, one of the other neurotransmitters. And the idea here is that you take more glycine, it's going to increase the methylation of catechols, and so that produces, I forget which neurotransmitter that produces, but in any case, I doubt it. But if you try it, you will see if it has an untoward effect. And it's not going to be a permanent effect anyway. If you try it and it makes you uncomfortable, or it increases your discomfort in any way, don't do it. Don't try it.
Yeah.
You still need glycine, but don't get it in a quick absorption form like the free amino acid. That may be that these people have genetic variants with relation to COMT, and maybe the glycine level affects it. But hypothetically, I would say your activities of glycine, of any kinds of methyltransferases, which we try to accelerate by adding glycine, it doesn't accelerate them. So it probably does not affect the action of COMT. My guess, my hypothesis.
Okay, thank you for that. And the last one was just more of a comment, and I wanted to share it with you. It says, I've been tracking a consistent dose-dependent biofeedback response with glycine. So at 6 scoops or 30 grams, sleepiness arrives smoothly, followed by a 15 to 30 minute neurological reset with no hangover. And at 50 grams, the sequence changes. I notice a darkening visual halo effect, then triggers the same 30-minute knockout. Both doses result in clear, clean-headed wake-up. So the identical reset timing suggests a clean metabolic clearance curve, likely supported by my restored eGFR of 78. I suspect the visual halo at 50 grams reflects a temporary shift in retinal microvascular tone before the CNS brake fully engages. Do you have anything?
eGFR? What's eGFR? That's usually glomerular filtration rate. That's usually a measure of kidney function.
I know. Okay, so it's of 78. So he suspects the visual halo at 50 grams reflects a temporary shift in the retinal microvascular tone before the CNS brake fully engages. I just didn't know. I didn't have any.
That may be. I'm not sure what it means by eGFR, unless they're just measuring that as because they're doing blood tests. The eGFR, glomerular filtration rate, is kidney function. Sometimes, if you take glycine supplementation, it will be interpreted as impairing your kidney function.
Yeah.
It doesn't. Glycine does not impair your kidney function, but what it does is it makes your liver and kidneys make more creatine and creatinine and urea. And what that does is, in a standard blood test, they use these algorithms in order to calculate. It's a calculation. It's not a measurement of your kidney function. It's a calculation based on the presence of these metabolites. And because you're making more of these metabolites, it looks like your kidney function is being compromised. So if you have any question, or if you're in a medical condition where your kidney function is being monitored, I would say stop taking glycine 3 days before every blood test.
I love that advice. Well, Dr. Brind, this has been amazing. Get your glycine, like we said, any way you can. Sweetamine, amino acid supplementation, bone broth, collagenous cuts. But thank you. Do you have any final words of wisdom? I'm so appreciative of your time and your continued interest in the research, despite the fact that you're retired and you're staying abreast, you're still willing to share. I mean, you're kind of retired. Retired from my particular position, but that's nice because this does give me an opportunity to continue teaching about it, because teaching is important. That's really how you learn. You really learn through teaching. And most of you who are teachers out there probably know that. I'm grateful for the opportunity. And, especially, that you provide with your, the audience, who is of people who are particularly tuned in to this kind of knowledge. It's a little different than teaching college. And in teaching college, if you have a lecture hall and you got 60 students in the classroom, sometimes you'll only get 2 or 3 who are really listening. And that makes it at least able for you to do the lecture. Yeah. If you ever walk into a room where nobody in the lecture room wants to hear it, all of a sudden you find you have nothing to say. Yeah.
So I know there's lots of people listening. Listening is a very active process, actually, contrary to popular belief. And I very much appreciate the opportunity to share what I know. And the more the merrier. And what I don't know, I'll hopefully find out. What many times it's what we don't know as a society, as an industry, if you will, in terms of the research industry. And medical research industry, it is an industry, and it's subject to a lot of the foibles and corruption aspects of it, as we all know. Money gets involved, and politics gets involved, and it's unfortunate, and just plain old forgetfulness about what the scientific process is. People think it's all about the data. The data is the last step to whether you confirm a hypothesis or have to modify a hypothesis. It's the key aspect of driving research in the right direction is asking questions, asking the right question. You start the scientific process by asking a question, and the answer that comes to you, that's your hypothesis, and then you test it, accept it, reject it. You test it mentally first, see if it makes sense. And if it ultimately makes sense in your mind, and then you have a means to test it, you try and test this testable hypothesis. If it's too complicated, you reject it. As I pointed out before, with this latest research on the connection between inflammation and depression, well, looks like common cause for different conditions like glycine deficiency seems to be more and more and more and more and more. We just keep adding. Once we found that glycine connection, it was like that magic piece in the middle of a jigsaw puzzle, and now all the pieces fit in. Well, the jigsaw puzzle just keeps getting bigger and bigger and bigger with all the research, but guess what? All the pieces still fitting into that same solution to the puzzle, into the big picture. So the picture becomes sharper even as it becomes bigger. And that's what really spurs me on, because you're onto something when all these things link up logically, when it seems more and more and more that something gets increasingly complicated and you throw your hands up, I can't figure it out. It's too many different markers and different genes and different gene products and different papers, and and you can spend your life just trying to read all of them and you can't get through it, and then you throw your hands up and say, what am I missing? That's a good question too. We know this, we know this, we know this, we know this, but it doesn't seem to give us the answer. What am I missing? Good question to ask to start you on a research journey.
Yeah. Well, thank you. Such wise words, Dr. Brind. And your podcasts are consistently our most popular. So people are listening. There's not just two people out there. There's a lot of people who are definitely benefiting from your knowledge. So thanks everyone for watching. And until next time, thank you so much, Dr. Brind.